I'm pleased to have Andrea Wolff here with us today. She has almost 20 years of experience with GLP and non-GLP in vitro metabolism and transporter studies at XenoTech. She transitioned from analyst to study director to manager for drug interaction studies across multiple disciplines, including drug metabolism, transporters, enzyme induction, and enzyme inhibition, and provided guidance to clients on the regulatory requirements for performing in vitro drug interaction studies. Andrea now manages several core service support teams that cover protocol writing, report writing, data processing, and project management. She is responsible for the quality and timelines associated with all contract study deliverables executed at XenoTech. It is my pleasure to present to you Andrea Wolff.
Thanks, Mike, for that nice introduction, and thank you all for joining me today as I present on understanding P-gp and BCRP assays and design. This presentation will cover the P-gp and BCRP assays that we offer here at XenoTech. I will briefly go over the test system qualification that we do before we would ever release one of these types of studies for a service. I'll also review some data to show test system differences and comparisons. I will then follow up with some data on IC50 versus KI, and then conclude with some data on clinical relevance. So the first type of assay that we perform or offer at XenoTech is the Transwell assay. This Transwell assay looks much like the diagram here, where there's two compartments. There's a smaller well that we call the apical chamber that sits inside a larger well that's called the basal chamber. And the cells are plated on a membrane that's in the apical chamber. We plate the cells, and then we allow the cells to grow on that membrane. They differentiate and polarize, forming a confluent monolayer with tight junctions resembling enterocytes of the small intestine. So then what we do is we measure the permeability of a compound in the basal to apical direction and in the apical to basal direction. P-gp and BCRP both pump in the basal to apical direction and restrict permeation in the apical to basal direction. So if a compound is a substrate of P-gp or BCRP, you should see a much larger transport of the compound across the cell membrane in the basal to apical direction compared to the apical to basal. So the data that you would expect to see for a compound that is a substrate, a P-gp or BCRP, would look something like the data that's shown here at 0 micromolar, where you have a higher permeability value in the basal to apical direction compared to the apical to basal, which is the clear bars that are down here at the bottom. And in this case, we're looking at P-gp data. The probe substrate is the digoxin. And so in the absence of inhibitor, we have a nice efflux ratio, which is the basal to apical divided by the apical to basal permeability. And when you add inhibitor, if it's a P-gp inhibitor or a BCRP inhibitor, you expect the basal to apical permeability to decrease, and usually the apical to basal permeability will slightly increase, and if there's complete inhibition, this usually will go to unity. The basal to apical and basal to apical and apical to basal permeabilities will essentially be the same, and the efflux ratio will go to 1. That's not always the case. It may be due to maybe endogenous transporters that are present in the test system itself, which is something we'll talk about later, but typically that is what you would see. And so you could determine an IC50 from the percent of control comparing to the 0 micromolar sample of inhibitor. For a substrate of evaluation, you would do the same exact thing that we did for digoxin here, where you would just evaluate, instead of digoxin, you would evaluate your test article in the basal to apical direction and the apical to basal direction to see if the B to A direction permeability is much greater than in the A to B. For P-gp, the test systems that we use are KCO2 cells and then MDCK2 cells that have been transfected with P-gp. And for BCRP, the test system that we use is the MDCK2 cell line transfected with BCRP. We use digoxin, as I mentioned, as the probe substrate for P-gp, and we use prazosin as the probe substrate for BCRP. The other test system that we use for P-gp and BCRP inhibition is the Vesicle assay. This assay is a little bit different. It uses basically it's human cells that have been transfected with P-gp or BCRP, and then they go through a process that inverts the plasma membrane of the cells. So now the transporter is on the outside. And since it's not a whole cell system now, when it goes through that process, it's no longer a cell system, complete cell system. It requires the presence of ATP to facilitate that reaction. So what you would do is you would add your probe substrate or your test article, whatever it is that you're measuring, to the vesicles in a media. You would add ATP, and then you would measure how much of that compound gets trapped inside the vesicles. You would separate the vesicles from the media by filtration, and you would lyse the vesicles, and it releases any of the compound that has been trapped inside the vesicles. You do this in the presence and absence of ATP, as I mentioned, and this is an example of inhibition data that you might get. In this case, we're looking at P-gp inhibition in vesicles, and the probe substrate is N-methylquinidine. We measure the accumulation in the vesicles in the presence of ATP and in the absence. And so the difference in these two values is the transporter-mediated uptake into those vesicles. And you would then measure that in the presence of multiple concentrations of inhibitor, and typically you will see a decrease in the accumulation of the probe substrate as the compound inhibits. And you can determine a percent of control value based on the 0 micromolar sample, and then determine an IC50 value from that. We don't use digoxin and prazosin in this test system because they don't work well in this test system. The ratio or the accumulation of the test article, the signal to noise ratio, is very low for those two substrates using vesicles. So instead, we use N-methylquinidine as the probe substrate for P-gp and estrone 3-sulfate as the probe substrate for BCRP. There are some advantages and disadvantages of these two transporter assays. In the Transwell system, you can measure permeability and bidirectional transport. You can't do that in the vesicles. So that's some added information that you can obtain from the Transwell system. It's also nice that these are cell lines that are stably transfected, and you can continually use them because they're immortalized, and they provide pretty consistent transporter activity as long as you don't go too high in passage number. The disadvantages of the Transwell system is that there is endogenous transporter activity present, and sometimes that can complicate the interpretation of your data. It's also important to note that there is some non-specific binding that occurs in this test system, and compound can actually bind to that membrane that is in the basal or the apical chamber. You can have some substantial reduction in recovery if you have some sort of non-specific binding. And if that binding is substantial, then it will affect your permeability values, and you may want to account for that in your calculations when you're determining permeability values. Since this is a whole cell system, you also are limited by cytotoxicity cytotoxicity. If you go too high in concentration with your test article, you could potentially start killing the cells, and sometimes that's hard to see visually in the transwell system. Because of that membrane, it's hard to see with a microscope. And so usually you want to measure cytotoxicity with some sort of endpoint like LDH release, because you don't want to confuse, cell death with inhibition. And sometimes that can kind of look similar. You can also measure TIR values or do some sort of permeability test post-assay, and that could also give you an indication if there was any disruption of the cell monolayer. And sometimes that's an indication of cytotoxicity. With the transwell assay, also the kinetics determinations are a bit complicated since you're looking at a bidirectional permeability. So there's a lot more that goes into those calculations when you're trying to determine kinetics. With a transwell assay, also we have a longer culture time, especially if you're using KECO2 cells. That requires a 21-day culture, and if you have some contamination or something and you lose those cells, then it takes another 21 days to grow up another plate to do your assay. And then also these assays require longer incubation times, up to 120 minutes usually. With the Vesicle assay, since it's not a whole cell system, the advantages to this is that you're not limited by cytotoxicity. You're more going to be limited by solubility. You can usually go as high as you can in terms of solubility. The other nice thing with vesicles is they're ready available. You can just thaw them and use them right when you need them. They have short incubation times, usually less than 10 minutes, and so that's really nice. You also have direct access to the transporter binding site since it's on the outside of the vesicle. Some compounds need to cross the cell membrane in order to interact with P-gp or BCRP, and that may require like some sort of other transport, an uptake transporter or something like that. You don't need that with the vesicle system, so you can just directly, measure kinetics or whatever you're trying to measure in that assay system and not worry about any other types of issues with the cell membrane, crossing the cell membrane. And then kinetics determinations are relatively easy. They're very similar to the approach that we use with microsomes and SIP assays. And it's just a much easier system to use when you're doing kinetics determinations. One of the disadvantages of using the Vesicle assay is that it's not very useful for highly permeable compounds. If a compound is highly permeable, it's just going to go in and out of that vesicle and it's not going to get trapped. And so you wouldn't be able to measure uptake, or the trapping, I shouldn't say uptake. You're not going to be able to measure the transport into the vesicle. You can also have some issues with non-specific binding, especially if it's getting trapped on the filter part of the plate when you're doing the filtration at the end of the assay. And then transporter activity can vary from batch to batch with the vesicles. The reason for this is uncertain, but it may be due to the fact that, with each different preparation of vesicles, the inside-out orientation of the vesicles may be slightly different. And so we recommend that you determine kinetics with each batch to ensure that you're doing accurate predictions of your kinetics. And I'll talk about that a little bit more later. So with each test system that we use here at XenoTech, we qualify it, and we think that's really important that it's done in order to optimize your assay conditions. As you know, or you may not know, the laboratory variability from lab to lab is quite different, and the procedures are slightly different, and so it's always important to characterize your test system in your hands in your laboratory. And so we do that with every test system that we use for service work here. And I'm showing today our qualification for the Vesicle assay, but we go through the exact same approach for all the other cell systems that we use for transporter work. So in this example, I'm showing the first step that we would do when we're qualifying a test system is we would do a time course determination. Now, we may do some preliminary assays before this to establish appropriate concentration to do these at, or do some literature searching just to figure out where to start. But typically, we're going to select a concentration that maybe we know a little bit something about, and we're going to do a time course. And in this case, because these are vesicles, the incubation times are rather short, and we went out to 5 minutes with N-methylquinidine in the P-gp vesicles and estrone 3-sulfate in the BCRP vesicles. And so on the left panels here are the transporter expressing vesicles, and then these are the control vesicles on the right with no transporter activity. And you can see that with the red bars, you see a nice increase in the transport in the presence of ATP. In the absence of ATP, it's pretty flat. And so you would want to choose incubation times moving forward where you're at the optimal incubation time. And so for P-gp or BCRP, we would probably select 2 or 3 minutes where it's kind of at the highest point before you start to see this like leveling off of the transporter activity. In the case of BCRP, we would select 1 minute in this case because that's where we saw the highest transporter activity. We saw a nice increase from 0.5 to 1 minute, and then it kind of plateaued out. So based on these results, we then move on to a kinetics determination to determine the Km value in that test system. We would use the time point that we determined during the first step, and then we evaluate multiple concentrations of the probe substrate to determine the Km value. We usually select these concentrations based on literature. There's usually data out there for probe substrates. For your test article, you may just select concentrations based on solubility, but you would evaluate multiple concentrations. In this case, I think we did 8. And you want to see this nice plateau at the top for a Km determination. And in this case, we evaluated 2 lots of vesicles. And the interesting thing that you'll notice here is that with the P-gp vesicles, the lot-to-lot variability wasn't very significant. And the Km that we got was 0.6 micromolar in both cases for lot 1 and lot 2. But for the P-gp vesicles, you can see that the concentration profile is significantly different for the 2 different lots that were evaluated. And when you use all the concentrations, the Km in the first lot was 13 micromolar, and in the second lot, it was 58 micromolar. So you're looking at approximately a 4 to 5-fold difference in Km values there. But if you also look at these data on the ED-Hofsey plot, which is always important to do, you can see that for P-gp, it's a straight line. But for BCRP, you're seeing this bend in the line. And that's suggesting that maybe you're having some allosteric interactions or differences in possibly binding sites to the transporter. And so we did a second calculation where we focused on the low concentrations where it would be the high affinity transporter activity. And that's what you see in this C panel here. And we're using just those low concentrations, and the Km in this case for the high affinity activity is 2.5 micromolar and 4.3. So it's really important for each different lot to evaluate that Km just to make sure that it's not changing from lot to lot, especially if you're going to use that in any sort of modeling later. So based on these data, we would then select a probe substrate concentration that's well below the Km value for our IC50 determinations. And the reason why we do that is, in theory, if there's competitive inhibition, at a very low probe substrate concentration using the Chain-Prusov equation, which is shown here, this value here would go to 1, and essentially the Ki equals the IC50. And so then you can make the assumption that the IC50 equals the Ki, and you don't necessarily need to do a Ki determination. So in this case, we do multiple concentrations of the inhibitor at a low probe substrate concentration below the Km. For P-gp, we evaluated verapamil, and for BCRP, we evaluated KO143. And these are our positive control inhibitors that we use in every assay for the Vesicle experiments. So the IC50 for verapamil is 3.9 micromolar basically, and the IC50 for KO143 is 0.1. So then the positive control inhibitor concentrations that we would use from assay to assay moving forward would be at least 10 times the IC50 value to meet the FDA requirements, as long as we're not limited by solubility. And so and we would expect at that concentration to achieve at least 50% inhibition. And so that's how we would select the concentrations for our positive control inhibitor moving forward. We also can determine Ki values with the Vesicle system, and you know, like I said before, can estimate the Ki from the IC50 value. So in this slide, I'm showing that we did estimate those values from those IC50 determinations in the previous slide. For P-gp, the estimated Ki was 2.6 micromolar. For BCRP, the estimated Ki for KO143 was 0.1 micromolar with all the concentrations included. At the high affinity range, the Ki was 0.0179 micromolar, which isn't actually that much different from the estimated Ki using all the concentrations. And then we experimentally determined those Ki's. And we perform these experiments similar to what you would do for microsomal incubations when you're evaluating CYP inhibition. We did multiple concentrations of inhibitor, multiple concentrations of probe substrate, and you can see from these slides that the data aren't pretty. They're not as nice usually as you would see for CYP inhibition type of profiles, but you can do it. And in lot 1, we just evaluated lot 1 for P-gp since those values, the Kms, were very similar. And the determined Ki was 2.9 micromolar, which is very similar to the estimated Ki, which again just shows that, and it was a competitive fit. So this just shows that, you know, you can estimate the Ki from the IC50 if you choose to do so. And then we also evaluated the inhibition, determined the Ki in 2 lots for a BCRP, since we did see those Km differences. So in lot 1, using all the concentrations, the Ki that we got was 0.1 micromolar, or 0.15 micromolar, which was pretty similar to the estimate. But also note that this fit is slightly different. It's non-competitive. And so the Chain-Prusov equation assumes that it's a competitive fit. So it's not exactly competitive, and so you wouldn't exactly expect this to be the same, but it is pretty similar. And then for the high affinity concentration range, we evaluated lot 1 and lot 2. In the case of lot 1, the fit was competitive, and in the case of lot 2, it was also competitive, but the values are slightly different. The Ki for lot 1 is at 0.079 micromolar, and for lot 2, it's 0.22 micromolar. And so the estimation of the Ki was spot on for lot 1, but for lot 2, it was slightly different, and that may be due to the differences in the Km values. So this slide shows 4 different compounds that we have evaluated internally with both the Vesicle test system and KCO2 cells. And we determined IC50 values for these 4 compounds. And some people make the assumption that since the compound has direct access to the transporter in the vesicle system, that typically the IC50 value is going to be lower. But that's not always the case. So in the case of valspadar and quinidine, we do see a lower IC50 value in the vesicle system. But in the case of verapamil and ketoconazole, we see actually a lower IC50 value in the CACO2 cells. And in all of these cases, the full difference between the IC50 values ranged from 0.4 to 10-fold in difference. And so this just shows that there are some differences in these two test systems. The reason for that is uncertain. It may be because you do have that interaction with the transporter, with the vesicle system. It is directly, you have direct access to it, whereas with the CACO2 cell system, permeability plays a factor in that, and so crossing that cell membrane may impact the transporter activity. And then also these are different probe substrates that we're looking at, and so there may be a slight different interaction of that probe substrate with the transporter itself, and so that may also explain the slight differences. So in the EMA guidance, it recommends evaluating two systems for PGP, and this just kind of confirms that it may be a good idea to evaluate using both the vesicle system and the CACO2 cell system to see what the differences are, if they're the same, or if one gives you a slightly lower value. And if you're want to take the conservative approach, you may want to use the IC50 value that you get from the lower, the lower IC50 value that you get in your modeling that you're going to do, or your calculations that you're going to do, just to be conservative and safe. Or you may want to evaluate both and see what kind of difference you would get when you model both IC50 values. So I kind of expanded this and looked at inhibitors in the University of Washington Drug Interaction Database. This is a very useful database and has a lot of information on transporter interactions. And I found 26 inhibitors in this database that use the same probe substrates that we use in the same test systems, and just compared the IC50 values between those two test systems and saw that there was a full difference ranging from 0.2 to 42. In most of those cases, the full difference was less than 10-fold, but there were several cases where it was outside that full difference. So again, this just supports the need for maybe evaluating more than one test system. All right, so jumping to clinical interactions, what does my in vitro data tell me? This table summarizes the criteria from both the, from both the EMA and the FDA. So this first column is the cutoff values for the EMA, and I'm just going to focus on P-gp and BCRP for this talk here. So they provide two different values. The first one is the P-gp and BCRP expression in the intestine. So you need to account for your highest dose that you're going to administer in 250 mils, which is basically, the dose with a glass of water, and 1/10 of that value. And you want to compare that value to your IC50 or your KI. And if your KI or your IC50 value is less than the value that you calculate from this equation, then you're going to want to have to do a clinical drug-drug interaction study. For the FDA, it's presented slightly different, but it's basically the same exact thing. And then the EMA also gives a systemic P-gp and BCRP consideration, and it's using 50 times the unbound Cmax. And if your KI or IC50 is less than 50 times the unbound Cmax, then you also need to do a clinical drug-drug interaction study. In the previous guidance for the FDA, there was a similar equation that was included in that guidance, but in the new one that came out in 2017, this equation was removed. And I think it's because in most instances, this equation up here is usually the higher of the two values, but it really also depends on whether or not you have a lot of compound that's bound or is unbound. And so we at XenoTech will ask for this information up front when we're designing a study for you. We'll ask you what the clinical dose is, the highest clinical dose, what the Cmax value is that you get at that dose, what the fraction unbound is, because we want to make sure that the concentrations that we evaluate for your inhibition experiment bracket those concentrations. We want to make sure that we're going high enough so that you can do these calculations and determine if you need to do a clinical drug-drug interaction study. We published a paper in 2015 after the EMA and the FDA recommended suspending the use of ketoconazole in DDI studies. They suspended the use, or they recommended suspending the use of ketoconazole because it causes liver injury or adrenal insufficiency. And they recommended some alternatives. They recommended clarithromycin, ritonavir, and itraconazole. And there wasn't a lot of published data out there on transporter inhibition of these compounds. So we performed IC50 determinations with 13 transporters, and that data is all presented in the referenced publication on this slide. So if you're interested in looking at that, feel free, or if you want to reach out to Michael or I, and we can send you a copy of that. But for the purpose of this talk here, I'm just focusing on the P-gp inhibition. So based on the IC50 values, we determined IC50 values for ketoconazole as well as those three compounds that the FDA was recommending that to replace ketoconazole with, and we determined these IC50 values in the P-gp vesicles. And then we took what we could find in the publications that are out there, what the clinical dosage is, the highest clinical dosage for those four compounds, compared that, and also the Cmax and fraction unbound. And then we did the ratio values, calculated those to determine if a clinical drug-drug interaction study would be recommended based on these IC50 values. And so the first calculation here is that dose in 250 mils. So it's using these values here divided by the IC50 value. And if that value is greater than 10, then that triggers the need for a clinical drug-drug interaction study. And as you can see, those values are well above 10. So in all these cases, you would predict that, or you would, based on these calculations, you would predict that there may be a clinical drug-drug interaction. And so you would want to do that type of study. And then we also did the calculation for the unbound Cmax over IC50, which is the systemic P-gp and BCRP that's basically expressed in the liver, and divided that by the IC50 and determined those values. And if it was greater than 0.02, then that indicated a need for a clinical drug-drug interaction study as well. And in the 3 out of the 4, that value was above 0.02, and with ketoconazole, it's pretty dang close. So in all of these cases, we would probably, if we were evaluating these compounds and they were just, normal drugs that hadn't been, they're not like actual drugs that are on the market, we would recommend doing clinical drug-drug interaction studies based on these data. So I think it's really important that these things are taken into consideration. In all these cases, these compounds are also 3A4 inhibitors. So you would not only have 3A4 inhibition, but you're also going to have P-gp inhibition. And so there could be definitely some implications that need to be taken into consideration there. So I also did a search on the drug-drug interaction database from the University of Washington to see how many drugs were P-gp inhibitors and were evaluated with ketoconazole as a drug-drug interaction study, and 26 compounds were listed. These 26 compounds all had inhibition greater than 20%. So these compounds are P-gp substrates. They were evaluated with ketoconazole to see if there was some sort of interaction, and there was. And only one compound was listed with no interaction, and that was lenvatinib. And then this table here shows 6 of those compounds with just huge increases in, huge increases in AUC when they were administered with ketoconazole. So the combined inhibition of 3A4 and P-gp in this case it creates a huge exposure of that compound, increases the exposure of that compound when ketoconazole is administered. And so this just supports the need for, evaluating P-gp and also being able to predict that type of clinical interaction using the cutoff values that the FDA and the EMA provide. So in summary, XenoTech qualifies our assays with the test systems that we use for service work before we ever release these systems for the service work that we provide to our clients. There are some variabilities between vesicle lots, and we saw that in the case of BCRP. And so we do recommend determining a KM with each lot of vesicles. KIs can be predicted from IC50 values, and so it's not always needed to determine a KI determination. Results between KCO2 and vesicles can be similar, but there are some notable variabilities, and so we do recommend evaluating 2 test systems if you just want to be very conservative. And then also I think we showed that the in vitro data are useful in predicting drug interaction studies. And that concludes my presentation today, and I just want to thank you all for listening in, and I think now we'll open it up for some questions, right, Mike?
Yes, indeed. Thank you very much, Andrea. While we wait for questions, I wanted to mention that information about our transporter services, papers, posters, other webinars, and more is available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. If you have any further questions regarding Andrea's presentation or any of Sekisui XenoTech's contract research services or products, please feel free to ask them in our post-webinar survey or contact us through our website. Okay, this wraps up today's session. Thanks to all of you for taking time out of your day to join us, and thank you to Andrea for her wonderful presentation. Have a great rest of your day.